The overall goal of this experiment is to utilize CRISPR-Cas9 gene editing to model calreticulin mutations in cytokine-dependent hematopoietic cells in order to study their oncogenic activity.
This method can help answer key questions in the hematological malignancies field, such as the biological function of driver mutations when they are expressed at physiological levels.
The main advantage of this technique is that it utilizes the CRISPR-Cas9 gene editing system to model and study the biological function of genes involved in cancer.
To anneal and phosphorylate the oligos, add the reagents to a total of 10 microliters in a PCR tube kept on ice. Set the program in the thermocycler. Use water to dilute the reaction after cycle is complete. To digest the Lentigard purovector, prepare 50 microliters of digester reaction. Then, start the digestion reaction at 55 degrees Celsius in a heat block. After 1 hour, add an additional microliter of BSM-B1 restriction enzyme and continue the reaction for another hour. To dephosphorylate the cut backbone, add 7 microliters of phosphatase reaction buffer and 2 microliters of phosphatase enzyme to the digested vector. Then, incubate the mixture at 37 degrees Celsius for 30 minutes. Purify the already cut and dephosphorylated backbone using a commercial kit. To prepare the ligation mixture, add 50 nanograms of digested backbone and other reagents to a PCR tube kept on ice, and adjust the volume to 10 microliters with water. Incubate the reaction at room temperature for 1 hour. Then transform STBL3-compatible cells with 2 microliters of the ligation mixture, and then plate on a lysogeny broth agar plate containing 100 micrograms per milliliter of ampicillin. After this, incubate the plate overnight at 37 degrees Celsius. Pick 3 colonies and inoculate them. Perform mini-prep for each culture, and sequence the oligo insertion site with a U6 promoter region primer. Seed HEC293 T cells on a 10 cm dish and leave it for overnight incubation. To prepare the DNA mixture for transfection, pipette 45 microliters of the transfection reagent to the DNA. Use a pipette to mix the DNA and transfection reagent carefully, and allow it to stand for 20 minutes. After this, add the DNA transfection mixture dropwise on the seeded cells. Gently swirl the plates and incubate at 5% carbon dioxide and 37 degrees Celsius for a day. Harvest the virus-containing supernatant after 24 and 48 hours by passing it through a 0.22 micrometer filter, and aliquot 1.6 milliliters of supernatant in each cryovial. Next, aspirate the supernatant and resuspend the BAF3 cells in media by pipetting the cells up and down several times. Add 500 microliters of the resuspended cells, 1.5 milliliters of viral supernatant, 4 microliters of polybrine, and 10 nanograms per milliliter of murine interleukin-3 in a 6-well tissue culture plate. Centrifuge the plate at 440 G for 120 minutes at 37 degrees Celsius. After centrifugation, transfer the plate to an incubator for overnight incubation at 37 degrees Celsius with 5% carbon dioxide. After this, spin the cells down at 440 G for 3 minutes and resuspend in 5 milliliters of fresh warm media in a 6-well plate. To select the Cas9 infected cells, first spin down the cell, and then resuspend in fresh warm media supplemented with 5 micrograms per milliliter of blastocidin after 48 hours of spinfection. Continue selecting cells for 9 days with blastocidin till all the uninfected cells are dead. Then transfer the resistant cells to a medium with 1 microgram per milliliter of blastocidin. Transduce the parental cells and the cells stably expressing Cas9 with PXPR011 reporter construct through lentiviral infection. Continue selecting cells with 2 micrograms per milliliter of puromycin for 3 days, 48 hours after spinfection. Infect the Cas9 expressing cells with single-guide ribonucleic acid using lentivirus. Then, use 2 micrograms per milliliter of puromycin to select the cells for 3 days, 48 hours post-spinfection. Spin the exponentially growing BAF3 cells expressing ectopic Cas9 and the single-guide ribonucleic acid of interest. Aspirate the supernatant and wash the cells with 5 milliliters of phosphate buffered saline. Spin the cells down and wash with phosphate buffered saline 3 times. Then count the number of cells using a cell viability analyzer. After this, seed cells in triplicates in 2 milliliters of fresh media without interleukin-3 in 6-well culture plates. Start monitoring and counting the cells every 2 days for 8 days. Representative flow cytometric data demonstrating Cas9 activity in parental BAF3 cell and BAF3 overexpressing Cas9 cells is shown here. Green fluorescent protein signal is reduced by 76% in BAF3 overexpressing Cas9 cells, indicating robust Cas9 activity. Whereas in parental BAF3 cells, the green fluorescent protein signal remains at 100%. Representative growth curves for cells demonstrating oncogenic activity of calreticulin are shown here. CRISPR-Cas9-mediated +1 base pair frameshift mutation was introduced in the exon 9 locus of calreticulin using M1 and M2 single-guide RNAs. Growth curves for calreticulin-targeted BAF3 thrombopoietin receptor Cas9 cells shows murine interleukin-3 independent growth. However, the growth curves for the other cell types do not show any cytokine-independent growth. Sequencing results confirm the on-target editing of endogenous calreticulin exon 9 in calreticulin-targeted BAF3 thrombopoietin receptor Cas9 cells, using the M1 and M2 single-guide RNA. The +1 base pair frameshift mutations are indicated in black.
Once mastered, this technique can be done in 2 weeks if performed properly.
While attempting this procedure, it's important to understand what kind of mutations in the gene of interest may be responsible for the phenotype that is desired.
Following this procedure, other methods to further characterize the mutation, such as its role in cell signaling or in protein-protein interactions, can be performed in order to further understand the mechanism by which it causes disease.
After its development, this technique paved the way for researchers in the field of hematology to explore the effect of gain-of-function mutations at physiological levels on cell transformation in BAF3 hematopoietic cells.
After watching this video, you should have a good understanding of how to utilize the CRISPR-Cas9 gene editing methodology to investigate the oncogenic activity of gain-of-function mutations in cytokine-dependent hematopoietic cells.
Don't forget that working with lentivirus can be extremely hazardous, and precautions such as working in a BL2+ facility should always be taken while performing this procedure.